Today you're going to learn about recombinant DNA. These will be your class notes. You should have a handout that looks just like this. So we're starting out with two vocab words. Eukaryotic cells. Eukaryotic cells are complex. And what makes them complex is that they have a nucleus. They have many other organelles too, but the nucleus is kind of the key here. It's like a container for the DNA, for the chromosomes. Your cells are eukaryotic cells. Plant cells are eukaryotic cells. Prokaryotic cells are simple. They have no nucleus. No nucleus. That's right, no nucleus. That's what makes them prokaryotic. So, bacteria are prokaryotic cells. Bacteria are prokaryotic cells, but they do have DNA. Okay? Bacteria have a single long chromosome. And they have little circles of DNA called plasmids. So bacteria have a single long chromosome plus circles of DNA called plasmids. So new vocab word for you. Plasmids are circular DNA. They are only found in bacteria. Now, plasmids have been found to be really useful in DNA technology. Scientists use them all the time. How do they use them? Well, here we go. Using a process called bacterial transformation, science can take out a gene that they're interested in. So they find a gene that they want, and they take it out. So using a process called bacterial transformation, scientists can take out a gene of interest. It's just a gene that they want to use. They take out a gene of interest and from one organism, like a human. Maybe they take a gene out of human DNA. So they take a gene of interest out of one organism and they insert that gene into a plasmid. Then they put the plasmid into a bacteria. Then the plasmid is put into a bacteria cell. This bacterium then contains a new gene, and it produces a new protein, because a gene, after all, is the code to make a protein. All right, and that is how we get human insulin. Okay, so it says, for example, plasmids are used to create bacteria that produce human insulin, which is a protein used by diabetics to control their blood sugar. Okay, if you don't know what insulin is, you should know it. Definitely a common medicine taken by many, many people. All right, so let's move on. This way, huge amounts of human insulin can be made. All right, and that is how we get our insulin now. All right, we used to get our insulin by grinding up the pancreas of cows and pigs. But now we have found the gene for human insulin. We cut that little gene out and we put it into a plasmid. We put the plasmid into the bacteria, and the bacteria make the human insulin. Well, how does that work? How exactly do we do it? All right, take insulin for example. The gene for making insulin is isolated from the DNA of a human by using something called restriction enzymes. Okay, restriction enzymes, which cut the human DNA at a particular place. This way, the gene for making human insulin is isolated. Next, a plasmid is removed from a bacterium. Scientists then use the same restriction enzyme to cut the plasmid at the location where the insulin gene is going to be inserted. All right. Cutting the plasmid is going to produce something called sticky ends, which you're going to see in a lab that we do. Okay, sticky ends are the two ends between which the insulin gene is going to be inserted. All right, I have a better definition, I think, of sticky ends, so I'm going to write it down. So these things called sticky ends. These are the kind of jagged ends of DNA that are produced when using restriction enzymes. Okay. All right, so that's the first part of the notes. Let's flip it over onto the back side and continue. All right, so once we have all of what's done on the front, we will say now. Okay, so now the plasmid is put back into the bacterium. Bacterium is just singular for bacteria. Okay, and it says the bacterium now contains the human gene for making insulin. The DNA in the plasmid of the bacterium is now called recombinant DNA. Okay, that's an important word, and here's the definition. Recombinant DNA is DNA from two or more sources. For example, DNA from a human, like the insulin gene, plus DNA from the bacteria. Okay, when you combine that DNA, it's called recombinant DNA. Now, let me show you. What I often show my students is whenever I talk about restriction enzymes cutting DNA, like cutting a gene out from a human, I often use a little symbol that looks like scissors. So whenever I show you this in class, that represents a restriction enzyme, okay? Restriction enzymes are enzymes that are naturally found in bacteria, and when scientists found out that they cut DNA, they thought, hey, what if we could use these restriction enzymes to cut DNA? And then maybe we could put some DNA together to make something new, and that's exactly what they did. So now I want to show you this little comic that I made. This is going to be page 2 of your notes, okay? So this is one way to transform bacteria using human DNA and bacterial DNA. So we're going to start out with a strand of DNA that is a human chromosome, and on this human chromosome there's a gene for human insulin. Okay, scientists take restriction enzymes that cut their gene of interest. This is their gene of interest right in here. Okay, the human insulin gene is the gene of interest, and the restriction enzymes are going to cut above the gene and below the gene. So when we do that, we say that we are going to isolate the human insulin gene. Okay, and we're isolating that human insulin gene from human DNA, of course. So that's one source of DNA. Okay. Next, we are going to take a bacteria cell, and it's got a plasmid, as you can see. We're going to take that plasmid out. So scientists will take the plasmid out of the bacteria cell. So remove one plasmid from the bacteria. Bacterium, single cell. Okay. And next, we are going to use our little molecular scissors. Okay, this is representing a restriction enzyme. And by the way, we're using the same restriction enzyme to cut the plasmid open as we used to cut the human DNA. You will see why that's so important. All right, so for our next little picture, we're just going to write that we are going to cut open the plasmid. All right, so we're using our restriction enzyme to cut the plasmid. We're using our restriction enzyme to cut the DNA chromosome. Okay, these little ends, now that we have opened up the plasmid, these little ends here are called sticky ends. Very scientific. Okay, so when you cut open the plasmid, you get sticky ends. Okay, so this plasmid, this is the plasmid cut open. Okay. Next, we're going to take that gene from above. We're going to take this gene and we are going to insert it into our plasmid. All right, so insert the human insulin gene into the plasmid. Okay. And last, we are going to put this plasmid into our bacteria cell. So now this plasmid has recombinant DNA. It's got bacterial DNA and it's got human DNA. All right, so now we're going to put the plasmid into the bacteria cell. And now the bacteria is going to read that human DNA as if its own, because DNA is a universal language. So it doesn't matter that it's from a human. The bacteria is going to read that gene as if its own. And now the bacteria will do whatever that gene tells it to do. And what this gene is telling it to do is make human insulin. So the bacteria can now make human insulin over and over and over. Okay, so that's all the bacteria is really going to be doing 24/7. Huge amounts of insulin will be made. Insulin is a protein, so the protein will be made at the ribosomes, and this bacteria will just be pumping out human insulin, which will be filtered and bottled and sold as medicine. All right, that's it. Thank you.